Hello students, hope you all are doing well. I'm Nitin and today we will discuss a new topic which is microtubules. It is one of the three main components of cytoskeleton which helps in maintaining the shape and internal organization of cell. Microtubules are rigid, hollow, rod structure of 25 nanometer in diameter. It is a dynamic structure that undergoes continuous assembly and disassembly within the cell. The main function of microtubules are cell locomotion, the intracellular transport of organelles, and the separation of chromosomes during mitosis. The structure of microtubules is a hollow rod shape composed of a single type of a globular protein called tubulin, which further made up of alpha-tubulin and beta-tubulin subunits. So as you are seeing in this picture, tubulin dimers polymerize to form microtubules, which generally consist of 13 linear protofilaments assembled around a hollow core. Microtubules are dynamic structures which can assemble and disassemble. The tubulin dimers with GTP bound to beta-tubulin associate with the growing plus ends, undergoing polymerization. Shortly after polymerization, the GTP bound to beta-tubulin is hydrolyzed to GDP. Since GDP bound tubulin is less stable in the microtubule, the dimers at the minus end rapidly dissociate. Well, dynamic instability can also occur when the hydrolysis of GTP bound to beta-tubulin takes place during or shortly after polymerization, which reduces its binding affinity for adjacent molecules. Growth of microtubules continues as long as new GTP-bound tubulin molecules are added more rapidly than GTP is hydrolyzed. So a GTP cap is retained at the growing end, and you can see that GTP cap is highlighted with a black boundary in the third image. However, if GTP is hydrolyzed more rapidly than new subunits are added, it leads to disassembly and shrinkage, as you can see in this picture. Well, microtubules also play a central role in mitosis. Various drugs that can affect the assembly of microtubules can be used for the treatment of cancer. For example, colchicine and colcimid are examples of experimental drugs that bind tubulin and inhibit microtubule polymerization, which further stops mitosis. Two other drugs, example vincristine and vinblastine, selectively inhibit rapidly dividing cells. Another anti-cancer drug named Taxol stabilizes microtubules rather than inhibiting their assembly. Such stabilization also blocks cell division. There are some proteins called microtubule-associated proteins or MAPs. The dynamic behavior of microtubules is regulated by microtubule-associated proteins or MAPs that bind to their plus ends. So as you can see in the picture, some of these proteins act as polymerases and accelerate growth by increasing the incorporation of GTP-bound tubulin. Further, depolymerases dissociate GTP bound tubulin from the plus end, leading to the loss of GTP cap and microtubule shrinkage or catastrophe can occur. There is another protein called class protein which can rescue microtubules from shrinkage by inhibiting disassembly and reinitiating the growth. Well, these opposing actions of MAPs can control microtubule growth and shrinkage as per the requirement of the cell. This is all about the microtubule structure, assembly and function. I hope this video will help you. If you like this video, please hit the like button and share your valuable feedback in the comment section. I'm grateful to Archana ma'am for her continuous support and valuable guidance in the development of cell biology video series. Many thanks to all of you for listening and appreciating our videos. Please subscribe to our channel, Mentorship towards Sure Abundance. Keep learning and keep growing. Thank you so much.